Bismillahirrahmanirrahim. My name is Dr. Mohammad Latif and I am your instructor of the subject Principle of Animal Life 2. Today is the lecture number 14 in which we will talk about recombinant DNA technology. What is a recombinant DNA technology? Recombinant DNA technology is a major DNA based tool that opens a new age for modern biotechnology. And the first concept for recombinant DNA technology came from Werner Erber's discovery of restriction enzymes in bacteria that degrade foreign viral DNA molecule. We'll talk about in detail about the restriction enzymes, and from this discovery geneticists learn to cut and paste DNA molecule, and novel restriction enzymes for cutting and pasting were discovered or invented. And the development of recombinant DNA technology was advanced by the collaboration of Stanley Cohen and Herbert Boyer in 1972. They also established the first company that focused on recombinant DNA technology. And with the help of this technology, a gene or multiple genes can be identified, cut, and inserted into the genome of another organism. Using this technology, the first drugs of medical biotechnology were produced, namely human insulin. Before this technology, the insulin can be obtained from goats, sheep, or from cattle. So with this technology, the human insulin can be obtained from bacteria. And what are the steps in recombinant DNA technology? There are 5 steps or process in recombinant DNA technology. Number 1 is the step in which we cut the desired DNA by restriction sites, and the other step in which we amplifying the gene copies by PCR. What is PCR? It is a polymerase chain reaction in which we make multiple copy of a single gene. Amplifying means copying or increase the number of genes. And the third step is the inserting the genes into the vectors. What are vectors? Basically, the vectors are used for insertion of genes. We'll talk about in detail in further slides. And then the transferring the vectors into the host organisms, at how these vectors are transferred into the host organism, and how we can obtain the products of recombinant gene. What is recombinant gene means? In which there is 2 genes attached to them, or 2 genetic material can be attached in a single gene. That's why it is called recombinant gene. So firstly, in first step, in which we isolate the gene of interest. Isolation of gene from chromosome is one of the basic steps or one of the prime job in genetic engineering, because Firstly, we have to identify the gene, that which gene is responsible for making a certain kind of protein which we want to make. Say for example, in case of insulin, if we want to make an insulin, firstly we must know what the segments of DNA in a human which make the insulin protein. So when we know the segments or a nucleotide sequence, we can isolate from the chromosome. And there are different ways in which we can isolate the gene of interest. Firstly, the isolation of gene from the chromosomes. Can be isolated from the chromosome by cutting the chromosome on the hanging sides of the gene. And there are special enzymes which is called restriction endonucleases are used to cut the genes. These are called molecular scissors because genes are made up of DNA and proteins, and there are certain enzymes which can cut the genes on a specific sites, and restriction endonucleases are the enzymes which cut the gene on the specified sequences. Or we can make chemical synthesis of genes. When we know the sequence of gene or the nucleotide, we can make in a laboratories, because we can make the molecules in a laboratory setting. So if we know the sequence of nucleotide, we can easily make in a laboratory. And the other one is making gene from mRNA. It is very common method of getting the gene. In this case, gene is synthesized in the laboratory from messenger RNA by reverse transcriptase enzyme, an enzyme which can make DNA from RNA. So when we use this enzyme, it can make DNA, and from DNA we can make protein, or we can make gene. And the other step is the molecular scissors. I have little talk about molecular scissors, which is called restriction endonucleases. The restriction enzymes are present in bacteria, and the bacteria use these enzymes for their own protection against viruses, because there are certain kinds of viruses which attack on bacteria and they kill them. So when a virus attack on bacteria, there are certain kind of enzymes present on the bacteria, which is called restriction endonucleases, which cut the virus DNA. And when the DNA of virus is cut, it is not able to kill the bacteria. So basically these are defensive enzymes, and these restriction enzymes cut down the viral DNA, but they do not harm the bacterial chromosome. They restrict the growth of viruses, so they are called restriction enzymes. Why they are called restriction enzymes? Because they restrict the growth of viruses. And Hamilton O. Smith isolated the first restriction enzymes in 1970. These enzymes cut the DNA at the very specific sites, and I am showing of DNA. The restriction enzyme cut the DNA at a specific site. These are the sites: AATTC. This is from 3' and 5' and to 3' and So it cut on the G nucleotide and the segment GCTTA are lost, and from the opposite side they cut from AATTC down G. So this segment, these are the specific segments in the target gene, which is called sticky ends. So these restriction enzymes cut the genes on the specific site, which is called sticky ends. EcoR1 is a commonly used restriction enzyme. It cuts double-strand DNA at the specific sequence of bases, so a gap is produced in DNA and a piece of foreign DNA with complementary ends. So when a foreign DNA with these ends can enter in this gene, it can easily join on this sites. Foreign gene with this end can easily join this segment of gene. So when they attach a piece of foreign DNA with complementary ends, the single strand DNA with complementary ends of the two DNA molecule is called sticky ends. Thus they can be bound by complementary base pairing. Therefore, the restriction enzymes help in the insertion of foreign DNA into the vector. What is vector? Vector will carry the desired gene. They are considered as the final vehicles that carry genes of interest into the host organism. And the most commonly used vectors are plasmid and bacteriophages. What are plasmids are? Basically, plasmids are natural extra chromosomal circular DNA molecule, and it is the most common type of vector. Plasmid, basically these are this can cause resistance against certain antibiotics, and it has a very significant role in bacteria. And the investigators discovered plasmid during their study of the sex life of intestinal bacteria, Escherichia coli. They carry genes for antibiotic resistance and fertility. And the other vector can be the DNA of bacterial viruses. The DNA of bacterial virus can also be used as a vector. For example, lambdaphage attached to a host bacterium. The recombinant DNA is released from the virus and enters the bacterium. And the recombinant DNA replicates, and many copies of the viruses are formed, and each virus is in bacteriophage clone contains a copy of the gene of choice, and from that copy we can isolate the gene. What is the expression system? You can see a human cell in which we isolate a gene for growth, human growth hormone, and there is a restriction enzyme called EcoR1 which cut the gene on a specific site. And then there is a bacteria in which there is a plasmid, and we isolate a plasmid from bacteria, and we also cut the plasmid to the specific sequence by EcoR1. This is basically the enzymes. Now the gene and the plasmid are come together, and because both have sticky ends or the ends which match their nucleotides, they attach with a complementary nucleotide, and in which there is a recombinant plasmid formed. And when these recombinant plasmid carrying the human growth hormone are inserted into the bacterium, the bacteria self or containing gene of human growth hormone will provide the material for its growth, and it will multiply the growth hormone gene, and from them we can isolate it. So this is basically the expression system in which we use bacteria for expression of our genes. One of the example is the human insulin. This is a human cell, human DNA. It can cut to a specific site by restriction enzymes. The insulin gene can be cut, and plasmid can also be cut by restriction enzymes, and the insulin gene and the plasmids are then DNA ligase seals human gene and plasmid, and this is called recombinant DNA. Why this is called recombinant DNA? Because it has one gene from insulin and the other is from bacteria. Now host cells takes up recombinant plasmid, and there is a cloning, means there is a multiple copy of this gene, and then these genes can be isolated, so we can make synthesis of a recombinant DNA. The bacterial cells are treated with calcium chloride. It makes the bacteria member more permeable. Now the bacterial cells take up recombinant plasmid. These bacteria reproduce and bacterial colonies are formed. Each new cell contains at least one plasmid. Therefore, each clone of bacteria contains the gene of interest. And the clone bacteria express themselves and make a product. The protein product can be separated from the clone bacteria, and the clone genes can be isolated from the bacterial clone for further analysis. So this is all about the DNA, recombinant DNA technology, and this recombinant DNA technology make wonders in genetic, in biotechnology. And in next lecture I will talk about the application of genetic technologies. In further video you will see about the process of genetic engineering, and this may helpful in understanding the topic.
One of the first genetic engineering experiments was conducted by Stanley Cohen and Herbert Boyer in 1973. They showed that the gene for frog ribosomal RNA could be transferred and expressed in bacterial cells. The first step was to construct the plasmid vector called PSC-101. This constructed vector contained a single site for the restriction endonuclease EcoR1 and a gene for tetracycline resistance. The restriction endonuclease EcoR1 was used to cut the frog DNA into small fragments. Next, the frog DNA fragments were combined with the plasmid vector that had also been cut with EcoR1. The sticky ends of the DNA fragments aligned themselves and the segments were joined together using DNA ligase. Some plasmids incorporate genes other than the rRNA gene, and some do not incorporate new pieces of DNA. The plasmids were then transferred into a tetracycline-sensitive strain of Escherichia coli and plated onto a growth medium containing tetracycline. The cells that incorporated the plasmid carrying the tetracycline gene grow and form a colony of cells. Some of these colonies consist of cells that carry the frog ribosomal RNA gene. The researchers then tested the colonies that form after growth for the presence of frog ribosomal RNA.